Shimika Kyusune. My name is Vivian. I am grateful for the opportunity to present my thesis via this platform despite the coronavirus situation. The title of my thesis is Apoptotic Effect of Natural Flavon Jaseucidine on MCF7 Human Breast Cancer Cells through modulation of HERC and P38 MAPK pathways under the supervision of Professor Namjuha. I'll be taking you through the background of the study, the objectives of the research, materials and method, results and discussion, and briefly I'd like to talk about cancer. Cancer is a typical disorder where malignant cells escape a balance between uh, proliferation and apoptosis, which makes them proliferate inappropriately. Like we know, there are several types of cancers, of which breast cancer is inclusive. Breast cancer has been reported as the ultimate cancer causing death in women globally. To combat this disease, uh, the treatment requires a successful balance between proliferation and apoptosis, which can also mean blockage of proliferation and induction of apoptosis. This has been recognized as a rational approach in combating this disease. For the purpose of this study, jaseucidine, a naturally active methylated trihydroxyflavone that is mostly abundant in Artemisia species, which is predominantly found in Korea, also known as suk, was used in the course of this. Quickly, I'll take you through my research objectives. My research objectives are 1. To corroborate the report that flavonoids commonly found in plants are associated with different biological and pharmacological activities. Secondly, to establish that natural compounds isolated from this plant have specific effects against cancer as compared with other methods of treatment, which includes chemotherapy or hormonal treatment. And lastly, to establish the apoptotic effect of this particular compound jasudin on MCF7 human breast cancer cell from the aspect of apoptosis pathway. Materials that were acquired in the course of this study includes the jasudin itself, a compound that was purchased from Chemfaces, human breast cancer cell, MCF7 in particular, purchased from Korean Cell Line Bank, human endothelial cells, EAH926, also purchased from Korea Cell Line Bank. To establish apoptosis, some experimental analyses were carried out, which include cell culture treatment, morphological assessment, cell viability, Western blot, flow cytometry, and of course, statistical analysis was employed in the course of this. This is a chemical structure of jaseucidine. Like I said earlier, jaseucidine is a trihydroxyflavone that has its hydroxy groups at position 5, 7, and 4, and its methoxy group at positions 3 and 6. It is mostly abundant in Artemisia species and has been reported to exhibit anti-allergic, anti-inflammatory, and apoptosis-inducing activities. Briefly, I would like to discuss about apoptosis. Apoptosis can be defined as a morphological and biochemically defined form of cell death. It could also be described as a programmed cell death. In the course of apoptosis, there's an active response of cells to the altered microenvironment and in turn leads to cell shrinkage, chromatin condensation, and DNA fragmentation. As we can see from this image, cells were treated with 10, 20, and 40 micromole of jaseucidine for a period of 24 and 48 hours respectively. Also, there was a control that was not treated at all with jaseucidine. From the image, at 24 hours, the cells were at full conferencing. As we can see, as the time and treatment increased, the number of seeded cells were decreasing, their morphology was changing, and the cells were shrinking. All of these processes are not visible cellular processes. Also, to confirm the cytotoxicity of this compound, jasucidin in the same dose and at the same time were used to treat the human endothelial cell HEH926, and from the picture as we can see, there are no significant changes or difference between the control cells and all treated cells regardless of their time. One of the other ways by which apoptosis is characterized is by examining the viability of the cells. Briefly, what do we understand by cell viability? Cell viability is a measure of the proportion of live and healthy cells within the population. Cell viability is detected using the entity assay, a yellow tetrazole solution, which later forms a purple formazan in living cells, is used. After some time, a solubilization solution like DMSO is added to dissolve this formation into a solution, after which it is then measured at a certain wavelength by spectrophotometer. From this result, the result shows that at 24 and 48 hours, the treatment of jasucidin decreased the viability of cells dose dependently as well as time dependently. Also, to also confirm this result on the normal EAH-HY96 endothelial cell, the same concentration and same time and same process was adopted in treating these cells. However, no cytotoxicity was observed in this cell and there was no significant difference in the viability of. Moving forward, one of the most important ways by which we can also examine apoptosis in the cell is by quantifying the expression of proteins associated with apoptosis as investigated in this study. From this result, we can see that there is an increasing expression of BAX, a protein associated with apoptosis, and also there is a decreasing expression of BCL2, as we can see in this study. A brief background about BAX and BCL2. They are both pro-apoptotic and anti-apoptotic proteins that contribute to programmed cell death or apoptosis. BAX is also a member of the BCL2 gene family, and both anti or pro-apoptotic regulators are involved in these different cellular activities associated with apoptosis. As we can see from the result, the ratio of BAX to BCL2 expression is increasing, which shows that which represents a cell death switch that determines the life and death of cells in response to apoptotic stimulus. An increasing BAX to BCL2 ratio decreases the cellular resistance of these cells to apoptotic stimuli, which leads to increased cell death, implying that jasucidin induced cell death in MCF7 cells. Moving forward, we can see that for PAP and for cleaved PAP, there is an increasing expression of cleaved PAP. PAP, briefly, is a nuclear DNA binding protein which can detect any DNA strand breaks. Once PAP is cleaved during apoptosis, then the DNA repair function is impaired. And once the DNA repair function of cells is impaired, it signifies apoptosis process. Also, we can characterize apoptosis by the cleavage of PAP. From this result, we can see that chasucidin treatment induced the cleavage of PAP. PAP is also an apoptosis. Worthy of note is that PAP is an apoptosis marker and a substrate of caspase. From the cleaved PAP to PAP ratio, we can also see an increasing expression in there. Some of the other apoptotic genes that were also examined in this study includes AKT, ERK, P38, and in the course of this blot, Western blot, beta-actin was used as control in all proteins. One of the functions of the AKT pathway is that it regulates cell growth, proliferation, survival, mobility, as well as inversion. It is a key regulator of physiological cell processes, like I have mentioned earlier. From this result, we can see an increasing expression of phosphorylated AKT, and also an increasing expression of phosphorylated ERK, as well as an increasing expression of phosphorylated P38. ERK, phosphorylated ERK, P38, phosphorylated P38, they are all molecular proteins that play major roles in controlling apoptosis, particularly ERK, which equilibrates the death and survival of apoptosis promoting genes throughout the MAPK pathways. Also, in figure 4b, as we can see from their ratios, the ratio of phosphorylated ERK versus ERK, and the ratio of phosphorylated P38 versus P38, there are also increasing ratio in their expression. Also, which implies that chasucidin modulates the MAPK signaling pathway to cause apoptosis in MCF7 cell, as observed from the result of these Western blots. And lastly, but not the least, to further establish previous findings, the cells were subjected to flow cytometry to determine the rate of cells undergoing apoptosis after staining with PI and annexin 5. From this result, we can see that chasucidin increased the apoptotic rate of the cells dose dependently as compared to the control cells. And also, we can observe that the cells underwent both early and late apoptosis. At a concentration of 20 micromole, 7% of early apoptosis was observed, while 17% of late apoptosis was observed. At a concentration of 40 micromole, about 36% of early apoptosis was recorded, and 40%, about 40% of late apoptosis was also observed. Like I said, this implies that chasucidin caused these cells to undergo both early and late apoptosis as the treatment dose increased. In conclusion, this study has been able to show chasucidin as a promising anti-cancer agent by enhancing apoptosis in MCF7 cells through the modulation of the MAPK signaling pathway. And to further establish this effect, in vivo study is required to corroborate these findings using the above concentrations. Here's an highlight of journals that I've been able to publish in the course of my master's degree, including this present research, which is being considered for publication in the Journal of Natural product risk. Thank you.